Okay, so I just wanted to walk through with you, some specifics with the Epithelial Tissue Lab. So when you go into Canvas and open up this assignment, this should be everything that you see. So if you click on this link for pages 95 to 101, it will open up a document that basically shows some pictures of our last textbook, which have some greater detail in them. And it might take a little bit for this to all get processed, but you should see pictures of the pages in the textbook that will help guide you as you go to complete this lab. Now remember, on the front side of this lab, it asks you some questions to do some matching and then some short answer questions. So I highly recommend that you refer to this information found in the old textbook, as well as your notes and your current textbook, to kind of help you to be able to make sure that you have the information you need. One of the questions asks to name the 4 major types of tissues and record their function. Well, look, there's a handy chart right here that helps explain all that. So utilize this because it will come in handy. I think the most applicable things are the pictures. And so if you see on the left-hand side, there's a drawing. On the right-hand side, it shows the actual pictures taken under the microscope. And then you see in the middle, they have everything labeled, so you can hopefully identify what's what. And then when you scroll down on this page, it then tells you where these samples were taken from or additional information. And then, like I said, it also talks to you more specifically about each of these types of tissues, so it really does come in handy when you go to do this lab. Now, as I mentioned in class with this lab, usually we get out the microscopes and a whole bunch of slides, and it can be really tricky stuff. It's not easy, but I know that you guys can handle it. Just do your best and use your tools to help you find what it is that you need. So instead of using the microscope, we're going to use this great resource online to help us find all the specifics that we need. And so if we go back to Canvas, you'll see that it says Histology Guide. So when you click on this, it will take you to this really awesome website that's going to show us basically exactly what it looks like when we look at these underneath the microscope. And as I mentioned in class, it also tells you additional information. So feel free to check that out, take a look at it, make sure that everything makes sense, and, continue to build on your knowledge. And so one thing I wanted to show before we go any further is we did record some things on our paper. So I was just going to put this up here so you guys could see. And I also will attach a picture on Canvas. It's a little bit blurry right now, but so this is kind of explaining the types of tissue that we most often think of containing these types of epithelial tissue, or the ones that specifically that we're going to look at with the website. And so I put for this first one lungs, because that's really what we think of with the simple squamous. But in this website, we're going to look at cornea. And so whenever I put an arrow, it tells you which specific slide to look at to kind of help narrow things down. So use that information to help you as you go. Another thing that you'll notice is you'll want to fill in these other blanks as well. So it says blank X. So you're just going to do your best to put the magnification, and I'm going to tell you usually that's going to be around 400x, so if you just put 400x, that would be fine with me. And then also it's going to ask for a function. Now I don't want the function of these organs, but I want the specific function of each of these tissues. And if you remember when we did our notes, that was part of the chart where it said this specific type of epithelial tissue has this specific function. So that's where I would go to get that information. And then if we scroll down so you can see the bottom, here's the final 2. And with the bladder, it's so fun, the transitional epithelium. But we talked about in class how it's kind of like a balloon and it can stretch when it's full. So that's when we call it extended. So you'll see it looks like those cells are stretched out. And then we go to the bathroom and we empty it, so they go whoop and they shrink back to their relaxed state. And it's really awesome how that transitional epithelium works in our bladder so that our body can not have to pee all the time. And then on the bottom, if you look here, I highlighted because I just wanted to make sure you guys didn't forget to do these things. But in each of these pictures, and we'll go through an example, but in each of these pictures, you're going to be asked to color code things so you don't have to label 5 million things. You're going to always label the apical surface and the connective tissue, but then also you'll see I have specific colors for specific components. So you just want to make sure that you're identifying all those structures as you look at all these different types of tissues. And then also don't forget to answer this critical thinking application. It's got 2 parts. Explain which one do you think, and then explain why you think that that's the case. So hopefully that should make sense as you go through and complete this activity. So I just wanted to talk you through, and I know that we did this example in class, but I'm just going to do it here for those kiddos that were not in class, or also to kind of talk you through it again, just in case. So we're going to take a look and draw this simple cuboidal epithelium that's found within the kidney. And so if we go back to the website, it shows... Sorry, I just have to find the right spot. If we go back, you'll see that I have the Epithelial Tissue Lab attached here, but that's the actual printout that you're going to hand in. So please don't do this digitally. You will hand in a hard copy of this activity. And then also, if you didn't take a look already, the epithelial tissue specifics, I made a video to help explain some of those particulars, especially if you weren't in class, but just kind of a review from biology and the notes that you guys took. So if you look at this Histology Lab again, so what's going to happen is on this website, and I got to go back to the beginning, so always click on this Chapter 2 Epithelium to go back to the guide. That's a great tool to use. But if you see, you can scroll through and take a look at all these different types of tissue. So really, it's this awesome resource because usually you'd have to go and exchange slides all the time. And like I said, I just wanted to keep you guys safe, so we're going to do things a little bit differently. So the first one we're going to take a look at is the simple cuboidal epithelium. And so we wrote down that we wanted to take a look at kidney, the slide MH0116. So that's this first one right here. So when you click on it, it basically shows you what it looks like under the microscope, this slide, and there's various samples. And the really cool thing is it gives you all these descriptions. So if you look, make sure you read through. It says some of these samples are taken from the ovary and some are taken from the kidney. Now you could look at either of these and should see similar stuff. But a couple things I wanted you to be aware of. If you look here at this little toolbar, it will let you pick specific things that you want to see. So it's really awesome because it takes a lot of the guessing work out of it. And then also along the bottom, a lot of times you can pick the magnification. And so like I said, mostly we want to go with 400x, which is the 40x that's shown here. So we want to zoom in on the kidney. So I'm just going to click on kidney just to show you how this works. And it's going to zoom in to this tissue to show us that this was taken from the kidney. But it's awfully hard for us to see particulars. So what we want to do is zoom in to what's called the renal corpuscles. So we're going to click on that and whoop, it zooms us in. So this is awesome. We look at these circles of cells, and that's what it's showing us, the simple cuboidal. So one of the big things that I mentioned in class is you want to make sure that I can see the type of tissue that it says on the lab. If it says simple, it should be just one layer. If it says stratified, it should be many layers. And then we take a look at the shape of the cells. Now sometimes it's hard to see, and we're going to have to use our imaginations, because if you look, it's really hard to see the cell membrane in these cells. But we're going to do our best to get all the particulars. So this one that's got an arrow pointing to it is an awesome example. And so this is what we're going for to draw on our lab. And so I'm going to try to bring my other screen in too so you can kind of see both at the same time, because hopefully that'll help us as we go to draw this. So bear with me a minute here. Okay. So here's my lab paper. I'm going to go to the part that says kidney. I also zoomed in. I don't know if you saw that, but I clicked on the 40x. So we're going to say 400x here for the magnification, and you'll do a little bit of extra research to figure out the function. So it tells us that we want to draw simple cuboidal epithelium. And it's really cool how it's arranged, because if you'll see, it's in this circle. So we were talking in class and I said it kind of looks like a donut. So go ahead and draw that. And my recommendation is to use a pencil, because we're going to add some color to this. And so hopefully you can kind of see, and you'll see my drawing's not going to be perfect, but I'm going to do as best as I can to show what this looks like. And then what happens is, this is showing my simple circle of cells, and sometimes that's how it is. They're not just going to be in this flat row, but that's a really cool thing. It lets us see how this really is inside of our body. And our kidneys have a whole bunch of tubes to be able to filter our urine, and so it's really cool how they work, and this is kind of showing us that. So we need to make some cells that are cube-shaped. So you're just going to go around and make some cube-shaped cells, and don't worry about it being perfect, but make sure that you can see it's one layer, or simple, and cuboidal. Those cells are cube-shaped. And then if you look on the bottom, it says we're going to use certain colors for certain things. And so the first thing that I notice when I look at this picture over here is I see these dark purple circles. So those are the nucleuses, and it says the nucleuses need to be red. So I'm going to color in and make my red nucleuses. And I'm just going to do a few because I'm limited on time here, but you should have these going all the way around your circle. It says the cell membrane should be green, and if you remember, the cell membrane is what goes around each of these cells. So it should be on all four sides of all of these cells that are in your circle of cells. Then it says the cytoplasm is orange, so we color in this space in between the nucleus and the cytoplasm, the nucleus and the cell membrane, orange. Oh, it's looking beautiful. Then we have to label some parts. It says we have to label the apical surface. The apical surface is what's exposed, so that means there's air, and it's white, so that's what's this in the picture. So that's what we have to point to. So when we go to our drawing, we want to point to the cell membrane that's exposed, and this is going to be how we label the apical surface. We also have to label the basement membrane, and the basement membrane is on the opposite side of the apical surface. So I'm going to make this blue line going around. This is the basement membrane, and it's said on the bottom that basement membrane is supposed to be blue. So there's our basement membrane. And then it says we want to label connective tissue. And so connective tissue, if you look, it's what's in between. And it's really just kind of blobby in this picture. And I don't care about you making too many details, but if you just want to kind of make some scribbles, that's fine with me. And then we'll put a bracket around it and say that that's connective tissue. So you just want to make sure that you're pointing to the apical surface, that's what's exposed. You're coloring the different components just like we did. We made the basement membrane blue. And then remember, in between the epithelial tissue and on the other side of that basement membrane, that's where that connective tissue is. We always have to have a connective tissue so that the epithelial tissue can have its blood supply. Hopefully that made sense. Feel free to reach out and speak up and ask if you have any questions. Use those pictures in the textbook as a guide for what you need to see. Like we said, they're not always going to be just a straight flat line of cells, but you're going to have to be creative. When you look at cornea, you're actually going to see that it's turned sideways. And so it's awesome, but read through the particulars that it gives on the side with the lab. Don't be afraid to use all those other components that it has, selecting the different views of the specific things you want to see and zooming in as much as you can to see all those particulars. And then, like I said, the interesting components that we really didn't get to talk about much are the goblet cells and the cilia, and those are on the two that are in the middle. So look at those pictures in the textbook to try to make sense of it. But the goblet cells secrete mucus, so they're always going to be towards the apical surface. And I'm going to try to have time to go here quick and show you. Let's go to the pseudostratified columnar with the trachea, so I can kind of hopefully give you a little example. Go computer, go! Pseudostratified columnar, and we want to do 136. And so there's different things you can click on. You can click on goblet cells. So what happens if we click on this? It'll zoom in and take us to those particulars. So this one is kind of goofy because it's upside down. So these are pseudostratified columnar. So basically all this